Hello, everyone, and welcome to today's webinar titled A CRISPR-Powered Universal Infectious Disease Assay. This webinar is part of the 2021 Microbiology Virtual Event, and I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Jumpcode Genomics. For more information about Jumpcode Genomics, go to www.jumpcodegenomics.com. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. And if you have any trouble seeing or hearing this presentation, just click on the Ask a Question box located on the far left of your screen. Now, as a reminder, the presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located at the top of the right-hand corner of your screen and follow the process to obtain those credits. Without further ado, I now present today's speaker, Jon Armstrong, the Vice President of Research Development at Jumpcode Genomics. For a complete biography of our speakers, please visit the Biography tab at the top of your screen. Jon, welcome. You may now begin.
Hello, my name is Jon Armstrong. I'm the Vice President of R&D for Jumpcode Genomics, and I'm really happy to be here today to talk with you about a assay we've been working on for the last year or so that leverages CRISPR and really uses CRISPR as an engine for a universal infectious disease assay. So, within the realm of next-gen sequencing and even metagenomic shotgun sequencing, one of the classic and historic problems is this needle in the haystack, where you know the things that you're looking for, but most of the molecules in the samples are uninformative. And ultimately, they mask the signal that you're looking for. And so we've been working diligently on a solution for this called the CRISPR-Clean technology. And I'll tell you a little bit more about how that works in the next couple slides. But ultimately, it enables scientists to focus on the signal and not the noise. So essentially, this is one way that our CRISPR-Clean technology can be employed, where it seamlessly integrates into the NGS workflow, where you have a sample prep, and then with that sample prep, you end up generating an NGS library, and then the CRISPR-Clean technology can then be employed at the end of the library prep to reduce the number of molecules that would be sequenced from non-informative areas. And then ultimately, as I just mentioned, you have the sequencing component. The technology works to increase sensitivity of detection. It's unbiased in the way that it works. It's a simple 4-hour total time workflow. Hands-on time is around 1.5 to 2 hours. And we see really efficient depletion of the regions or genes or fragments that we're targeting. And at the end of the day, it allows researchers to access lower input amounts and sample types than you might have been able to previously do with a depletion technology that happens up front and works to reduce your total RNA amount. And so this is how CRISPR-Clean works. So ultimately, we use Cas9 and guide RNAs. We design guide RNAs to target the areas of the genome or the transcriptome that we don't want to end up taking up real estate on the flow cell or the sequencing machine. We can use Cas9 and guide RNAs complexed in an RMP and then digest those library fragments that the guide RNAs are designed to. Once those fragments are digested, then a simple PCR amplification is used, and since the adapters that are ligated on the ends of those fragments in the NGS library construction process, since now those fragments are in two pieces, no PCR product can be formed from those fragments, and we end up with a material that's highly enriched for the areas that are informative, and the material has been depleted of the areas that are non-informative, and I'll show you data from that as we move through the talk today. And so 2021 for us has been a focus on RNA products. We have a product for transcriptomic analysis, would be bulk RNA sequencing. We have one for microbiome metatranscriptomics, and I'll show you some early proof of concept data that we generated using the metatranscriptomics kit. We have a single cell analysis product as well. And then finally, the one I'm going to be talking about today is our infectious disease surveillance product and kit for that assay. So on the left here, there's a lot of words, I understand. You can find all of this on our website, of course. Let me give you the take-home. The take-home here is that in metagenomic samples, things like nasopharyngeal samples, saliva samples, fecal samples, or even microbiome samples, this is a complex sample that's going to contain a mixture of bacterial and human cells and bacterial and human ribosomal RNA and total RNA, or other RNA species, as well as viral RNA and fungal RNA. And so these are complex samples. Those complex samples and the complexity of that means that really you have a needle in the haystack problem. When we use the CRISPR-Clean technology to deplete and effectively eliminate the bacterial and human ribosomal RNA from these samples, we essentially facilitate the identification of lower expressed transcripts and biologically relevant transcripts. And so the metatranscriptomic kit that I'll show some proof of concept data from and also our CRISPR-Clean Plus kit that I'll show data at the end of the talk is designed towards human ribosomal RNA species that are listed here in the table on the right, and to the sum or the superset of ribosomal species across 200 plus bacteria. And if we have a recommended library type for a post-PCR depletion using the NEB-Next Ultra 2 directional RNA kit, but also in October, we'll be releasing a full solution that bundles together the depletion reagents with an RNA library kit as well. And so here's the workflow for that bundled solution. It will be the CRISPR-Clean stranded total RNA prep with ribosomal depletion or CRISPR-Clean Plus in the case of metagenomics or universal infectious disease detection, where we're putting the depletion of the ribosomal RNA instead of after the library process. We're actually depleting fragments that originated from ribosomal RNA between the adapter ligation and final PCR amplification. It's really seamless integration, makes the library prep extremely easy. The hands-on time total is only 1.5 hours for this additional step, and really allows a competent tech to be able to move through the library process and depletion with numerous sample types within about a day and a half. So first, I'd like to present some early proof of concept data that we generated using our metatranscriptomic kit that depletes human ribosomal, but that ribosomal is designed for mouse and rat as well, plus pan-bacterial ribosomal. And this was early data that we generated that helped us to understand the path forward for a universal infectious disease assay. As mentioned here, this is on clinical fecal samples that we worked on this with. And so when we take a look at the y-axis, what we're looking at here is 3 fecal samples with and without depletion, and then looking at the percentage of ribosomal RNA that's remaining in those samples with and without depletion after sequencing. And the y-axis shows this, where when we have a control sample that's non-depleted, of course, the total ribosomal species in a stacked bar plot is going to make up 100% of the ribosomal species. But then after depletion, you can see in each of these samples, we're reducing the ribosomal content to below 10% and in some cases below 5% in these 3 sample types. And so we're seeing efficient removal of bacteria and human ribosomal RNA, and that includes gram-positive and gram-negative bacterial samples. And as I mentioned, the samples processed were clinical human stool samples. And next, we wanted to take a look at, okay, if we've now taken and removed these fragments from a library, and ultimately these fragments now don't take up real estate and form clusters on a flow cell, that area on the flow cell is now available for other informative fragments to form clusters and ultimately sequencing reads. what kind of increase in sensitivity of detection would we obtain with that? And you can see here that when we take a fecal sample that's been non-depleted and sequence that fecal sample, at least in these three samples, and then look at the bacterial species detected, shown here on the y-axis, you can see that we're detecting somewhere around 35 to 40%. I'm sorry, 30 to 40 species of bacteria are detected in these samples. But when we apply the Jumpcode depletion technology, the CRISPR-Clean metatranscriptomics kit, we see that that increases to somewhere around 130 to 140 bacterial species detected. And so thus, we're gaining a large increase in detection, 3 to 4-fold increase in the number of bacterial species identified with the CRISPR-Clean tech in these early experiments. And so what this essentially equates to is an increase in sensitivity of detection of the bacterial species in these samples. And so thus gave us the drive to continue to move forward in development and take a look at what we might be able to do with human samples with COVID spike-ins. So the next short slide of data I'm going to show you is some proof of concept experiments that we performed using a contrived sample that has COVID spike-ins titrated. And so there's a lot of information here, and I'll walk you through this. So essentially, we did a titration, a COVID spike-in titration where we went from 1% spike-in into total RNA to 0.0001%. And that's the percentage fraction against the total. So we took 1 nanogram of total RNA lung, and we spiked in Staph aureus and COVID-2, the Wuhan strain from Twist Biosciences. And then in the upper left-hand graph, what we're simply looking at here is we're simply looking at the raw read alignments to the Staph aureus genome and the COVID genome with and without depletion. And so you can see here that without depletion, we have about 20,000 reads or so aligning to that, aligning to those two genomes. And we have a certain proportion of genomes as well that we're seeing for that alignment. And then when we then look at the post-depletion, you can see that that goes up considerably to over 180,000 raw reads. And so we're gaining a large fold increase here in reads that are aligning, raw reads that are aligning to those two genomes. And in fact, if you look at the ratio, it's more of a qualitative measure here, but you can see the ratio of Staph aureus to COVID RNA in the samples is retained through the depletion procedure. So there's some, an unbiased methodology that's happening. In the right-hand graph, you can see that, of course, as we decrease the titration of the COVID control into the sample, and we compare the fold change between the mock to depleted samples, you can see here that as we decrease, of course, our fold change is going down, but still, even at the lowest spike-in amount, at 0.001% viral fraction, we're still retaining around an 8-fold increase in percentage of read alignments that are going to the COVID genome from mock to depleted. And so based on these results, which are extremely promising, we wanted to move forward to really build out a full solution for a universal infectious disease assay. And so next I'm going to move through multiple slides of data that shows that we generated for contrived and nasopharyngeal sample data. So we're 3 nasopharyngeal samples from COVID positive patients using our CRISPR-Clean plus stranded total RNA prep with depleting human ribosomal RNA and pan-bacteria. As I mentioned, our ribosomal RNA guide set is designed to human, mouse, and rat, and also effective on multiple eukaryotes. But don't want to be confusing here. These are human samples that we're working on with those 2 guide sets in our plus kit. And so a quick note on the technology before I dive into the data here. When we think about infectious disease surveillance, and certainly the data that I'm going to show you here and the methods that we're employing to generate this data, we don't think about infectious disease depletion technology as a replacement to amplicon sequencing or qPCR for simply COVID detection. Our technology is really meant for when you want to know more or take a deep dive into a subset of, let's say, a subset of positive samples, maybe even higher CT value positive samples, for the potential of CTs that show up as negative, but still the person has some showing a sickness. And so in this case, you'd want to be able to take a look at things like co-infection or typing of the viruses, even human host response signals, and potentially unknown pathogens. So these are the areas that we believe shotgun metagenomics paired with the Jumpcode CRISPR-Clean technology really takes on its real value, is wanting to do a deep dive into what these samples have in them and being able to look at multiple compartments of information. And so that's the data that I'm going to show here. So first, just the basic study design. We have 3 nasopharyngeal COVID positive samples. These were a CT of 19, 26, and 30, and I'll show that on each of the slides. So we have a high viral titer, an obvious positive, a medium viral titer, and a low viral titer, which in some cases under certain assays may have been called a negative. And then we applied the CRISPR-Clean plus depletion with the HMR ribosomal RNA and panbacterial ribosomal RNA guide sets. We also performed a mock depletion control for each of these samples. And then they were sequenced on a NextSeq 2000 at 2x100 base pair reads. I'll mention later that we downsampled those to 28 million reads per sample, the lowest number of reads for any of the samples. We downsampled every sample to that. And then we analyzed the data with multiple pipelines in order to understand composition of the samples through ID-Seq, the strain typing using EasyBiome, and also using in-house custom pipelines for genome depth and breadth of coverage, as well as the ribosomal RNA depletion rate. And I'm going to show you data today that covers each of these types of analysis, and I'll mention and point out each of these as we move through and what pipeline was used. So first, using our in-house pipelines to understand rates of depletion for ribosomal RNA, with our contrived sample, which essentially was made up of 50% human RNA, 50% bacterial RNA, and a 0.1% COVID spike-in, you can see here that the mock sample, that the ribosomal RNA originating from human is approximately 75% in the orange bar, and that's the y-axis is the percent of total RNA. And then the percent of ribosomal RNA coming from bacterial is approximately 25% in the mock sample. When we use just the bacterial guide set to deplete this sample, you can see that we completely obliterate the bacterial ribosomal RNA, and the component, that percentage of that component now is less than 5% in the total. Conversely, if we use the human guide set only to perform depletion on this sample, we then completely obliterate the human ribosomal RNA, and thus that to less than 5%, and thus reducing that compartment down. And so, as you can see here from the graph on the left, both of these guide sets work very, very well and efficiently individually. And then when we mix them together, we would expect the same results, and that's shown here on the right, where we're simply now looking at just the percent of RNA, ribosomal RNA depletion from either human or bacterial. When we mix the guide sets together and we're using those, using those guide sets at the same, using those guide sets on the same samples, you can see that in the mock, we have, and in sample 370 and 404, we have approximately 85% ribosomal RNA total prior to depletion, and in the 396, we have around 60%, and then after depletion, we're seeing less than 10% across all 3 of those samples when we deplete the human ribosomal and bacterial ribosomal. And I'll remind you that with this result, what the effect of this is that now we essentially are have the ability to take all of these reads that previously, 75% of these reads that previously would have been soaked up by ribosomal fragments, and we can now reassign those reads to an informative portion of this microbiome sample or nasopharyngeal sample for us to better understand what the components are in there, what the host response is, what other pathogens may be present, and also to be able to strain type the COVID in these COVID positive samples. And so first, let's talk about composition of the samples. What we've done here is we took a mock depleted sample, a mock sample for each of the three. So we have our high CT to low CT from left to right respectively here. And what we did is we took the mock sample for each of these three. We then defined the top 10 organisms based on reads per million using IDSeq. Reads per million is on the y-axis here. And then we simply looked in the depleted sample and said, for these same organisms that made up the top 10 in the mock, how many reads per million do we have of those? And then we can develop a fold change for reads per million between mock and depleted. And I'll just make a blanket statement across all of these samples. For this range of CT values, we're seeing anywhere between 4 to 10 plus fold average increase in fold reads per million going to the top 10 organisms in these samples. And just as a sanity check, wanted to take a look here at some of the composition of these several of these organisms in the top 10. And when we take a look at Malassezia restricta, it's one of the most abundant species of the human skin microbiota, and we see it in all three of these samples. Also, when we look at Cutibacterium acnes, you can see that this was also seen in all three of the samples, and it is also a part of the commensal flora of the skin and can be found in the mucosa of the nose. So these are nasopharyngeal samples. Not surprising that we're seeing these in the top 10 organisms from a reads per million standpoint, but also good that we're seeing these in the top 10 organisms. So this is checking out here. What's interesting, though, is then when we look at the reads per million for COVID in these samples, the mock sample prior to depletion actually did not show COVID based on a reads per million in the top 10 organisms in that sample. Very low viral titer at a CT of 30 plus. But then when we look at the middle CT and the low CT samples, we can see that COVID now starts to show up in those top 10 organisms. And in fact, in the middle sample, when in the mock it's showing up as number 4, and then when we deplete that sample, we get a 10-fold increase in reads per million that's going to the COVID genome. And we're seeing about a 2.5 or 3-fold, somewhere between 2.5 and 3-fold increase in reads per million to the COVID genome, and even the low CT or high viral titer sample. These are very interesting results in the sense that we have the ability, using this technology in shotgun metagenomics, to go in an unbiased and also hypothesis neutral way, start to look at what the compositions of these samples are, while also being able to obtain the designation of the COVID genome. I also mentioned that there's the potential of unknown pathogen identification. And in sample number 2, you can see here that we've identified Staph aureus, and in fact, in the depleted sample, we're gaining 10-fold increase in reads per million to the Staph aureus genome. And as we know, Staph aureus, it's a usual member of the microbiota of the body. It's frequently found in the upper respiratory tract, but it can cause respiratory infections. And so if someone were to potentially present with a fever or other types of problems or sickness that could be potentially thought of as COVID, but in this case, it might have been found that it could have been due to Staph aureus. Obviously, this is a COVID positive sample, but I'm just trying to define an example here where, because we're able to identify the other organisms in these samples, this is a very good way to be able to uncover co-infection organisms or unknown pathogens. And now let's focus our attention away from the composition to actually the COVID genomes. So this is in the contrived sample where we have spiked in the SARS-CoV-2 control from twist at 1.1% of the total RNA. And you can see this is simply meant to show the delta between untreated to depleted using the CRISPR-Clean Plus kit. You can see here in the mock, the genome coverage is in orange. It's not very easily identifiable and very low. If we were to just deplete the human ribosomal RNA, such as a product that's currently on the market now, we see an increase in coverage. But when we use the Jumpcode CRISPR-Clean Plus kit, you can see that we have an enormous increase in coverage over the COVID genome, a tenfold increase in SARS-CoV-2 genome coverage in this contrived sample, and thus simply an increase in sensitivity of detection of the COVID genome, and also an increase in base pair coverage of the COVID genome, which allows one to call variants in the genome and thus to perform strain typing. Then when we look at the nasopharyngeal COVID positive samples from the 3 patient samples with same thing here, CT, high CT on the left to low CT on the right. So it would be low viral titer to high viral titer, respectively. You can see here, we're not only looking at the average depth of coverage per base that's shown on the y-axis over the COVID genomes between a mock and depleted sample, but we're also showing the breadth of coverage. And that's seen at the top of each graph next to the legend designated by the letter capital B. And so you can see here that in the the high CT sample on the left, that with no depletion, simply just shotgun sequencing, the average coverage is approximately 0.12. And so whereas with depletion, the average coverage is 1.985. And this represents around a 16x increase in coverage over the COVID genome with depletion, even at a CT of 30 plus. And concomitantly with the genome, we can see that in the mock, we have a 9% breadth of coverage of the genome at 1x coverage or greater. And then when we deplete that sample, that breadth of coverage of the genome at 1x coverage or greater increases to 93%. And this represents a 10.3x increase of breadth of coverage of the genome. So with a mock sample here at a CT of 30 plus, you can see that depending on the sample number and what the low level of noise might be of alignment to the COVID genome, an average coverage of 0.12 could potentially be in noise. But certainly with an average coverage of 1.9 and a breadth of coverage of the COVID genome of 93%, even at a CT of 30.38, this would not be in the level of noise and could be seen as a positive. The same topology and increase is seen across the range of CTs, and one of the greatest ones that we see is in sample 404, where we're seeing a 10x coverage in coverage depth between the mock and depleted, where we're going from a 1.7 to a 15.5 average coverage per base between mock and depleted. So a large general increase across the coverage genome here. Just as an orthogonal validation, we wanted to look at, and sorry, let me go back here. There we go. We wanted to look at these same types of coverages using the ID-Seq analysis platform. The previous graphs were shown using our in-house custom analysis platforms, but using the ID-Seq, we're seeing much of the same type of information show up with sample 370 here. We're seeing an increase in coverage depth from 0.2x to 2.2x. This is at a depth of coverage of 10x or greater. And so we're requiring 10 reads overlapping a base to count this as covered or not. And so we're seeing a tenfold increase in coverage there. And then a breadth of coverage at 10x or greater, moving from 11.7% in the mock to 42.4% in the depleted. And then Same type of movement here as we move to the middle sample, sample 404, 1.7x to 13.5x increase in average coverage at 10x, and a 71.7 to 99.4% increase in breadth of coverage. So as I mentioned before, a universal infectious disease assay, be great to be able to understand composition of the sample. I've shown you data on that. Understanding of coverage of the COVID genome in this case, since these are COVID positive nasal pharyngeal samples, we've looked at that. Also, understanding the strain type of the COVID genome. And here's data, I'm sorry, keep pushing the wrong button. Here we're showing data for variant detection of from these clinical samples with a CT of the middle and the low CTs. We're able to divine the variant in those. In the low CT, we see a predominance of the Netherlands variant there, and in the middle CT, predominance of the US variant there. The EasyBiome variant detection algorithm, so this is data using EasyBiome, as I mentioned before. It uses single nucleotide variants in the alignments, and sample 370 simply did not have enough in there to pass the filter. However, we're continuing to improve our methodology and increase the sequencing depth slightly from 28 million reads, and we fully expect that we'll be able to detect the variants and the single nucleotide, the variant of that COVID genome and the single nucleotide variants in that sample in the future. And then finally, the host response. And so what we've simply done here is we're looking at human gene read counts for these nasopharyngeal samples. And we've cut off noise at 100 reads here, or 100 reads per, sorry, we've cut off our counts here at 100. So that's why the X and Y axis, the beginning number is at 100. But ultimately, we wanted to take a look at the linearity of the signals between mock and depleted samples in the human transcriptome, or the host genes in these samples. And you can see here that the linearity of human gene expression is retained with depletion for the medium and the low viral titer samples. Essentially, the high viral titer sample had so much virus in it that it took up quite a bit of the total RNA space in there, and thus the sequencing for the human transcriptome here is essentially a low input RNA sequencing experiment. And the linearity decreases with these high viral titers for just the reason that I mentioned. But ultimately, this information and this linearity provides potential for future development around host response, especially in samples that show medium to low viral titers. And those things around host response could be biomarkers around host response in response to treatment, or even in response to severity of infection. And so, in summary, with the CRISPR-Clean plus depletion, all non-human species receive around a 5 to 10-fold increase in reads. The coverage of the SARS-2 COVID genomes increase across all CT values, and the sensitivity of sequencing is equivalent to PCR to detect the presence of the COVID pathogen. The discovery and assessment of co-infection or primary infection species is available using this technology, since the depletion affords a large increase in sensitivity. And also, there was an effective variant detection across, or COVID variant detection across a range of viral loads. And so final thoughts is that shotgun approaches will eventually become the standard, and they will become an unbiased tool for infectious disease testing. And our Jumpcode CRISPR-Clean depletion makes shotgun metagenomic sequencing a viable tool for infectious disease surveillance. And Jumpcode is supporting this early transition to an unbiased pathogen detection. So thank you today for taking the time to listen, and happy to take any questions.
Thank you, Jon, for that informative presentation. And we will now start the live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just use that Ask a Question box located on the far left of your screen. So let's get started. It looks like we already have some great questions coming in. Jon, here you are using CRISPR guides that target human and bacterial ribosomal transcripts. Could you target other things as well, and how would that affect the sensitivity of the assay?
Yeah, sure. So we absolutely can target. We can actually target any transcript or genomic region that, as long as we meet our filtering criteria for uniqueness and GC content and other things. But ultimately, within the realm of a universal infectious disease assay, one area is if there's less interest in the host response or the human RNA data that I showed at the end of the presentation, the red XY plots, then you could go after additional genes there outside of just the non-coding ribosomal transcripts. You could also go after the highly expressed poly-A ribosomal genes, as well as the highly expressed poly-A mitochondrial genes. Probably make up about 20% of the reads that we're seeing there. And then you could also go after things like actin, GAPDH, other highly expressed genes, and probably recapture another 50% of the sequencing data to reattribute or reassign to the bacterial and viral compartment. And so, and what that, essentially the effect of that is in sensitivities, that raises the sensitivity of detection, raises the genome coverage, as I've shown here, that when we deplete the ribosomal, we're increasing genome coverage. And so it helps to increase sensitivity of the assay.
Thank you so much. And I want to, again, thank our audience for these questions. We have another question coming in. How many CRISPR guides are you using? And is there an upper limit to the number that you can use? Sure.
So in this assay, we have approximately 13,500 guides that we're using, since we're designing and targeting to 200 plus bacterial species. That takes up a lot of guide space. So we have about 13,500 in this assay. We've tested up to 600,000 guides within a genomic depletion environment, where we're trying to deplete regions, non-informative regions of genomic DNA from non-human organisms. But at this point in time, we haven't really been able to reach what that limit is. Like I said, we've tested 600,000 guides, and it works effectively and efficiently. And we'll just continue to test, and at some point in time, we may find that upper limit, but right now we don't quite know where that is. But 600,000 guides gives us a lot of target space.
Absolutely. Thank you so much. And I want to thank you for your presentation today and for your time, Jon. Would you like to provide any closing remarks for our audience before we go?
I think I'm okay. Thank you for asking, and thank you for listening to the presentation. And if anyone has any questions for me, they can either track me down through the folks here on the line, or likely find me somewhere on the interwebs. So.
Well, again, I want to thank you for your time and for your research. And before we go, I also want to thank our audience for joining us today and for their interesting questions. And I want to thank Jumpcode Genomics for sponsoring today's webinar. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast will be viewed on demand for 2 years until September 9th, 2023. LabRoots will alert you via email when it's available for replay. And we encourage you to share that email with your colleagues who may miss today's live event. Until next time, everyone, take care and stay healthy. Bye-bye.